Hello everyone, my name is Akram Vasighizaker, the PhD student in computer science from the University of Windsor, and today I'm going to present predicting intercellular signaling from single-cell RNA-seq data via graph convolutional neural network. The main goal of this study is discovering and analyzing intercellular signaling network. As we know, intercellular communication allows cells to communicate with each other in response to changes in their microenvironment. So this ability to send and receive signals is essential for the survival of the cells. And so identifying and quantifying intercellular signaling pathways has become a major problem in computational biological studies. Link prediction is one of the key problems in graph theory. Also, with the rapid advancement of single-cell RNA sequencing technology, researchers are becoming more interested in uh, inferring cell-cell communication from single-cell RNA-seq data. A graph convolutional neural network could be a possible solution to solve the link prediction, to solve this problem. It means we can transform gene expression matrix into a graph representation, graph structure, in a way that nodes can represent cells in the graph and edges represent cell-cell interactions. Well, previous studies work on different link prediction approaches, and they just assume some specific assumption about the link between cells, and they show a high performance for only some specific problems. On the other hand, subgraph-based methods solve this issue, and in this work, we inspired by the subgraph-based methods. We present the pipeline, including 3 main steps that I'm going to explain in the steps in more details. The first step is data preprocessing, including basic filtering, normalizing, log transformation, scaling, which we use just as standard preprocessing steps for single-cell RNA-seq data in this step. The next step is to create cell-cell graph from the single-cell data. For this, we just use preprocessing single-cell data to create similarity matrix S, and then we extract adjacency matrix from the similarity matrix to extract cell-cell graph. And the final step is to predict interaction between cells using graph convolutional neural network. This is the main step of our pipeline and includes some sub-steps. First one is extracting explicit features using a pooling layer. In this step, we use information gain to extract informative and important features as explicit features. And also we use cell-cell graph to extract subgraphs and create a node embedding on the graph. And also we did some node labeling to extract some latent features in a way that we can use a hashing function to assign some integer values to the nodes in a subgraph, extracted subgraph. And the results of all of these steps would be attributed graph X, and it would be input for the graph convolutional neural network. To evaluate our proposed method, we did some comparative analysis with latent methods, and we did some experiments on 6 RNA-seq datasets, including human and mouse pancreas tissue. And the result shows that our proposed methods outperform the other latent methods, including node-to-vec, line, deep-fog, and spectral clustering, and the performance in terms of area under the curve, it's near to 1. It's almost 0.99%. Also, we calculate other performance metrics, including accuracy, precision, recall, and FS score, and all of the data sets, and it shows almost approximately 99% for all performance metrics as well. And these figures are provided ROC curve for only two of the data sets, and the true positive rates against false positive rates. And it's noticeable that the proposed methods surpasses other approaches because the curve is closer to the top left corner, indicating better performance in both figures. In more detail, the exact value for false positive rate and true positive rates for all uh, methods against our proposed methods is showing in these charts. And it's showing the lowest false positive rate for the sexical, implying that there is a lower probability that a sexical will predict non-interacting cells as interacting cells. It means when there's not have any interaction between cells, the chances of inaccurate predictions, I mean the cells interacts, are minimal. So sexical performs best in predicting actual interactions. In a brief, the key ideas used in this work is just exacting local subgraph from a given network around the target nodes to capture locality, and also we build cell-cell communication using cell-cell similarity metrics. We employ information gain in the pooling layer as an approach for like coarsening the graph attributes while preserving the global structure of the input graph. And also we capture the latent as well as explicit features of the graph constructed from gene expression profile of individual cells. And the key references used in this study is as follows. And thank you very much for your attention, and I'm open to answer any specific, any potential questions.